guys, today's video is about the microbial metabolism. The microbial metabolism is the means by which a microbe obtains the energy and nutrients it needs to live and reproduce. Microbes use many different types of metabolic strategies, and species can often be differentiated from each other based on metabolic characteristics. All living cells depend on biochemical reaction to maintain homeostasis. All of the biochemical reactions in an organism are collectively referred to as metabolism, which is of two basic types: the anabolic and the catabolic. Anabolic is the reactions that build new molecules, while the catabolic is a reaction that break down molecules. All organisms, prokaryotic or eukaryotic, need to build the molecules they need and find the energy to do so. Most biochemical reactions are part of a series of reactions referred to as a metabolic pathway. In this pathway, you can see that it takes multiple reactions to make end product. Pathways can be catabolic or anabolic. Each reaction is catalyzed by its own enzyme. Now, let's go to the enzyme basics. Almost all biochemical reactions are catalyzed by a specific enzyme, proteins that accelerate the rate of a reaction without being changed themselves. The need for enzymes provides a way to control or regulate biochemical reactions. So reactions won't occur unless the enzyme that catalyzes the reaction is present and active. Enzymes also will lower the activation energy. So in this figure, the reactions won't occur unless the energy requirement is met. So the enzymes physically binds substrates. So the biochemical reaction can be controlled by changes in enzymes activity, which can be influenced in several ways. So as you can see in the diagram, the first diagram shows the temperature. Well, the reaction occurs more rapidly as temperature rises. So as long as the enzyme is active, heat can denature the enzymes. So that's what will happen. Then the next diagram is the pH. So enzyme structure depends on pH. The pH affects charge of the R group's protein structure. Then the last one is the substrate. Reactions occurs more rapidly as it rises. Now, let's go to the enzyme denaturation. Enzymes are polypeptides that retain their ability to function only when folded properly. So, changes in temperature, pH, or can disrupt amino acid interaction, causing the protein to unfold and become denatured. Now, let's go to some enzymes required cofactors. So, it can be a metal ion, vitamin, or other non-protein. If the cofactor is organic, it is called a coenzyme. So, enzyme is inactive without cofactor. So, this illustration shows the enzyme inhibition. The inhibitors bind enzymes in one of two ways: the competitive inhibition and the allosteric inhibition. So, inhibitors can bind reversibly or irreversibly. So, in the end product of metabolic pathways are important reversible enzyme inhibitors. So, the inhibit first enzyme in pathway, turning the pathway off. The low inhibitor is equal to pathway on. Then the high inhibitor is equal to pathway off. Can be a competitive or allosteric inhibition. Provide an important way of regulating an end product levels. Now, let's proceed to the ATP production. So, the ATP or the adenosine triphosphate preferred source of usable energy for all cells. Breaking bond of third phosphate release ideal AMT of energy or amount of the energy. So, bond is easily broken. This is why organisms convert food energy to ATP energy. So, how is ATP produced? So, in most organisms, energy from a food source is converted to energy in ATP by glycolysis, followed by one of two processes, the fermentation and the respiration. First, let's go to the glycolysis. So, glycolysis is the catabolic pathway by which sugar, such as glucose, are broken down into 3 carbon molecules of pyruvic acid. Now, in this process, the release of energy will yield 2 ATP per glucose. So, also transfers high energy electrons to NAD positive to yield to NADH. Now, let's go to oxidation or reduction. So, much of the energy in food molecules such as glucose is captured as high energy electrons by electron carriers such as the NADH and FADH2. So, when a molecule receives or gains electrons, it is said to be reduced. So, a molecule that gives up electrons is said to be oxidized. So, now let's go back to the process of producing ATP. So, the fermentation is the next. So, ATP production begins and ends with a glycolysis in organisms that ferment. Fermentation is all about recycling NAD positives so that glycolysis can continue. So, the NADH is oxidized to NAD positive by reducing pyruvate to lactic acid. So, you can see in the illustration. So, there are different fermentation products, so which is the fermentation in product, the ethanol, the acetic acid, lactic acid, propionic acid and carbon dioxide, then acetones and butanol, glycerol, citric acid, methionine, and sorbose. So, next is the third one, which is the respiration. So, after glycolysis, energy in pyruvate and NADH is used to produce much more ATP by respiration. So, here includes the Krebs cycle. So, it breaks down the pyruvate to 3 CO2, energy captured as A negative by NADH and FADH2. So, next is the electron transport. E negative from a NADH and FADH2 used to produce H positive gradient. Then, the third is the chemiosmosis. The H positive gradient used to make ATP. So, first, what is the Krebs cycle? So, it is the process, it is a cyclical metabolic pathway catalyzed by enzyme in the matrix or mitochondria. So that is the Krebs cycle. It requires 2 C-acetyl groups connection to coenzyme A. So acetyl coenzyme A. 3C pyruvate plus CoA or coenzyme A. Then 2C acetyl coenzyme A plus CO2 is the result of a Krebs cycle. Now let's go to the electron transport and chemiosmosis. So the electron transport and chemiosmosis is occur in the mitochondria of eukaryotes and at the plasma membrane of prokaryotes. So the oxygen is usually the final electron acceptor. But other molecules can play this role in anaerobic respiration. Now let's go to the lipid and protein catabolism. So lipids and proteins can also be used as source of energy to produce the ATP. So different amino acids enter glycolysis or the Krebs cycle at various stages. Fatty acids are broken down to acetyl group and feed into the Krebs cycle. Now, let's go to autotrophic process. So all organisms depend on autotrophs. Autotrophs can produce organic molecules from CO2, an inorganic carbon source. So all heterotrophs require an organic source of carbon, organic molecules directly or indirectly. and come from autotrophs. So the source of energy for autotrophic processes can be the light or the chemical. So in light, photoautotrophs can carry out photosynthesis, while chemical, chemoautotrophs that use various molecules as a source of high energy. So what are the light reactions of photosynthesis? So the electrons energized by sunlight. So fuel the synthesis of ATP through electron transport and chemiosmosis. So much like a respiration. So ultimately reduce NAD P positive to NAD pH. So the ATP and NAD pH provide energy to fuel production of sugars in the dark reactions. So what is the dark reaction? So involves an anabolic pathway known as the Calvin-Benson cycle. So endergonic reaction of this pathway are fueled by ATP and NAD pH from the light reaction. So resulting sugars can be used as a source of energy to build others organic molecule. So that is how the metabolism of a microorganism processes work. So now you can see the illustration, which is the summary of the energy metabolism.
That's all for today. Thank you for watching.